All right. So we have talked about the different methods of drug design, computer-aided drug design. Okay, and then we have talked about the softwares that are used for those different types of drug design. So let us quickly get into what is the criticism for drug design, okay, or the disadvantages of CAD. So it has been argued that highly rigid and focused nature of rational drug design suppresses serendipity and drug discovery. So we have those two methods, ligand-based drug design and structure-based drug design. Okay, and many researchers they argue that because these are very stringent methods, we might lose out on the serendipity that is there in drug discovery. These are very focused methods, right? रिसेप्टर का स्ट्रक्चर पता होना चाहिए या ligand का स्ट्रक्चर पता होना चाहिए and in doing all of those focused experiments we might lose out on the serendipity. So serendipity as drug discovery is lost. That is a criticism that is done for computer aided drug design. Because many of the significant medical discoveries have been inadvertent, the recent focus on rational drug design will limit the progress of drug discovery. So that is what is people who support serendipity in drug discovery say. Furthermore, the rational design of a drug may be limited by a crude or incomplete understanding of the underlying molecular processes of the disease it is intended to treat. Okay, so for instance, हम लोग बोलते हैं receptor structure based drug design or ligand based drug design. Okay, and uh, and then what we do is we talk about how well the ligand is binding to the receptor in structure-based drug design, or we try to predict what would be the kind of receptor structure that is associated with this particular set of ligands, and then we'll try to design new devise new ligands which will bind very nicely to receptor structure, right? So what happens in this? We do not talk anywhere till now. We have not talked about what will happen when the ligand binds to the receptor. उसके बाद में there are some secondary messenger systems, right, which are activated. There may be some conformational changes that may be induced in the ligand or by in the receptor. Okay, and that might have an effect on the overall therapeutic effect of the drug. So we are not talking about those underlying molecular processes. Okay, that may be responsible for the disease, either physiologically or pathologically. So this is one big disadvantage of computer-aided drug design, that we do not look into the underlying molecular processes of the disease, either physiological or pathological. हम लोग सिर्फ यही देखते हैं कि whether the receptor, whether the target is involved in the physiology or pathology of the disease or not. That's it. That is a disadvantage of drug design. Okay. Having said that, let us move on to talk about physicochemical descriptors. So here we'll talk about the importance of physicochemical descriptors and then we'll generate using website Mole Inspiration. एक website है which is called as Mole Inspiration and we'll have an assignment based on that. Okay, so out of the 15 lectures that we'll be having for computer aided drug design, 3 lectures would be for assignment. Okay, so let us talk about the physicochemical descriptors first, their importance first. Okay, now before moving even a slide ahead, let me just talk in brief. Now, we have heard of many molecules which enter into clinical trials, right? Now, what is the process? The first thing is we have to design new chemical entities. Okay. Once the new chemical entities are designed, they enter into preclinical trials. Preclinical trials and animal studies. Okay. From preclinical trials, they enter into clinical trials and human studies. And then they are approved by the FDA. Okay. They are approved via NDA, New Drug Application by the FDA. Okay, and then they are called as drugs. So these drugs are then marketed. Okay, these drugs are then marketed, and when they are marketed, phase 4 clinical trials are done. Okay, so clinical trials phase 1, 2, and 3 happen over here, but phase 4 clinical trials happen after the drug enters into the market. Right, now what is observed is NCEs, so many NCEs are designed and discovered and they are subjected to clinical testing. Okay, and they show good efficacy. Then they move on to clinical trials. And what is seen is in these 3 phases of clinical trials, So many molecules they fail. Okay, many molecules they fail in the first three phases, that is phase one, phase two, and phase three of clinical trials. And the reason behind that is usually, you know, they will be having additional toxicity, or they may be having additional metabolism, or maybe their absorption distribution may not be good. Okay, or maybe their excretion is happening very quickly. Okay, so what are we talking here? We are talking about A, T, M, E, T, right? We are talking about the physicochemical properties A, D, M, E, T. So that means if most of the drugs are failing due to adamant properties, then physicochemical properties which are directly responsible for those adamant properties are to be blamed. Barabar. And hence there is an important and hence there is a very urgent or absolute need for studying adamant properties during the drug design study stage as well. Okay, so adamant properties. Medicinal chemist may just discover a drug, may just design a drug, and say that I have designed a new drug. But what if it is failing for adamant properties? So that is where pharmaceutical chemistry comes into the picture, right? So I'd explained to you all the difference between medicinal chemistry and pharmaceutical chemistry, right? So medicinal chemistry talks about drug design and drug synthesis and drug preliminary evaluation, whereas pharmaceutical chemistry will talk about their physicochemical property optimization as well. Pharmaceutical chemistry will talk about hit identification, hit to lead optimization, and lead optimization, right? So lead optimization, hit to lead optimization is done by modifying the physicochemical properties that will have a direct effect on the adamant properties. Okay, absorption, distribution, metabolism, excretion, and toxicity. Now, this is how physicochemical properties are very, very important. Initially, many researchers, many big pharma companies, who invest heavily in drug discovery and drug design, they used to miss out on this particular set of physicochemical property descriptors or physicochemical properties. And then their molecules used to fail in the clinical trials. Okay? So later on, they observed physicochemical properties are very important, and they started optimizing those physicochemical properties in the initial phases itself. Okay, and this is how physicochemical properties have gained importance. Okay, so this is the importance of physicochemical properties or physicochemical descriptors that need to be studied. Okay, so in the next slide, we'll see a list of physicochemical properties that should be ideally studied. So the first thing is log P, O by W. If you all remember, in our previous lecture, we talked about QSAR, right? QSAR, we said that SAR used to deal only with changes in the substituents and their effect on the activity. But QSAR deals with changes in the physicochemical properties and the effect on the activity, right? So QSAR study करने के लिए भी physicochemical properties are very very important. That is another important role of physicochemical properties. Okay, so octanol water partition coefficient log P O by W. We have blood brain partition coefficient log BP. Now log P O by W we know is a parameter that is related to the oil solubility versus water solubility, right? So octanol represents the oil phase, water represents the aqueous phase, and log P O by W gives us the oil to water solubility ratio, right? Then blood brain partition coefficient or log BB. Log BB talks about how nicely the molecule will permeate across the blood brain barrier into the CNS. Okay, then log S is aqueous solubility, and aqueous solubility we know is very important. Those who are from pharmaceutics know this even better. So if we have high log P, that means it will have low log S, right? So high log P indicates high lipid solubility. High log S indicates high water solubility. So they are inversely proportional, right? So if we want to make a formulation, both of these are important. Lipid solubility is also important and water solubility is also important, and they have to be balanced, right? Then CACO2 cell permeability or MDCK cell permeability. Now these two are cell lines, CACO2 and MDCK. These are cell lines which are predictive for intestinal absorption. Okay, we know that in our intestines we have similar to blood-brain barrier, we have a barrier in intestines. The intestinal barrier is much better, I mean, but much easier to be overcome as compared to the blood-brain barrier. We all know that from our physiology studies, right? But intestinal barrier ka permeation is what is talked about by CACO2 cell permeability and MDCK cell permeability. Okay, so how good the drug will be able to permeate across the intestinal barrier? That is what is CACO2 and MDCK cell permeability. And skin permeability. We know that we have transdermal route of absorption or of administration, right? So skin permeability is a factor that comes into the picture under such circumstances. All right. So skin permeability is log Kp. Then free energy of solvation in water, free energy of solvation in octanol, free energy of solvation in hexene. These are all different parameters that talk about the solubility in water or octanol or hexene. Okay. Then we have human oral absorption. Now this is a predicted value wherein we talk about how easily the drug will be absorbed. Okay, when it is taken orally. So this is analogous to Lipinski's rule of five, right? Lipinski's rule of five that we had studied. So if a drug is following all those rule of five, then it will be possibly orally bioavailable. That is what we said in there. So human oral absorption talks about the percentage oral absorption that may be possible. So if a drug is absorbed to more than 80%, then it is very nicely absorbed. Okay. If the drug is absorbed to greater than 50%, then it is okay. It is, I mean, okay absorbed. If it is less than 50% or rather less than 20% absorption, then that means the drug is poorly absorbed. So these are all predicted human oral absorption values. Then we have CNS activity. So, some of the drugs, we want them not to enter the CNS. For example, antihistaminics. We do not want the antihistaminics to be entering into the CNS because once they enter into CNS, they will cause sedation. Right. So those molecules which are not supposed to be CNS active should not have CNS activity. And this value, this physicochemical property helps us to predict what would be the possible CNS activity. So if it is say something like +2, that means it is highly CNS active. +1 is CNS active. 0 means it is not having any activity in the CNS. It can have -1, -2 values as well. So those values would indicate that this given drug or the drug that we are trying to design will not enter into the CNS and have some CNS activity. Okay, so we know all of those side effects, right? Let's say we have sedation, we have convulsions, we have dizziness, we have nausea. All of those are CNS related side effects, right? So if we want to evade all of those CNS side effects, then the molecule should not have any CNS activity, right? So that is what is meant by this physicochemical property. Then polarizability. Now this is an indication of how polar the molecule is. And ionization potential. This is the energy that is required to remove the outermost electron that is present in the particular atom of the molecule. Then electron affinity. It is nothing but the affinity for the electrons. Okay, so these are all electronic parameters. Then number of likely metabolic reactions. Now this is important. Many times the drugs fail in the clinical trials because they are, highly metabolized. So if we can predict the number of metabolic reactions that can happen for the given drug in the drug design stage itself, then that will be good. Right? The probability of the molecule failing the clinical trial stage would be reduced. So this is an important parameter. and prediction of binding to human serum albumin. Now we know that albumin is one of the blood proteins, plasma proteins, right? So if a molecule binds to to a plasma protein, then that part of the molecule, that concentration of the molecule will be not useful unless and until it is in the bound form. The moment the molecule is released from the plasma protein, it becomes free and it becomes available to bind to its receptor, right? So this is an important parameter, especially for understanding what kind of dose would be required. So, for instance, drug like warfarin is almost like 99% plasma protein bound. That means only 1%, less than 1% rather, is freely available for having its effect, right? Therapeutic effect. But so that means we have to administer large amount of warfarin. We have to give a dosage of large amount of warfarin so that even though 99% is plasma protein bound, that less than 1% which is freely available is going to cause its therapeutic effect. Right. So this is an important parameter to be checked. And maximal transdermal transport rate. So this is again related to transdermal route of administration. Then number of amines. So these are the number of non-conjugated amine groups. Their value should be in between 0 and 1. Amidine. Number of amidine and guanidine groups. Their value should be ideally 0. Okay. Then number of acids, number of carboxylic acid groups that may be present in the given molecule. So this should be ideally 0 to 1. The number of amides, the number of non-conjugated amide groups that are present in the given molecule, they should be ideally 0 to or 1. The number of rotors, that is number of non-trivial, non-hindered rotatable bonds, यानी कि C single bond C. Okay, so they should be in between 0 to 15. 0 होंगे तो भी चलेगा, but maximum 15 single bonds होने चाहिए, rotatable bonds होने चाहिए. Then this is hash RTFG, so that is number of reactive functional groups. Okay, so reactive functional groups, like for example, we have so many of those functional groups, like for example diazo. Okay, diazo is a functional group which we know can react with multiple proteins. Then penicillins, we know they react with certain amino acids that are present in our body and that can lead to allergic reactions. So the number of reactive functional groups that are present on the given molecule should be checked. Okay, because these specific groups, you know, these groups can lead to false positives in the HTA, that is high throughput screening assays, or they may lead to decomposition, or they may lead to reactivity, or they may lead to toxicity when they are taken in vivo. Okay, so this value, number of reactive functional groups, should be ideally 0 or maximum it can be 2, but definitely not more than 2. Then CNS predicted activity. So it can be -2 for inactive, +2 for CNS active. Okay, just excuse me, this is repeated. Okay, so CNS predicted activity. So -2 indicates that the molecule would be inactive in the CNS, whereas +2 indicates that the molecule will be active in the CNS. Then molecular weight. The molecular weight of the molecule, we have studied this in Lipinski's rule of 5 as well, right? It has to be less than 500. That is what Lipinski and his colleagues had stated. But this parameter has been again studied in detail, and now it is known that molecular weight up to 725 is okay. Okay, so a drug should have a molecular weight in between 130 and 725 ideally. Then dipole, this is dagger. Okay, this symbol is called dagger, dipole dagger. So computed dipole moment for the molecule, and that should be in between 1 to 12.5. Then SASA. SASA is the solvent accessible surface area in square Armstrongs using a probe. तो ये SASA measure करने के लिए ना एक hypothetical probe with a radius of 1.4 degree Armstrong is placed near the molecule. Okay, and the solvent accessible surface area is computed. So this value should be in between 300 and 1000. And we have fossa. Fossa is nothing but the hydrophobic component of the sasa. अभी ये sasa जो है, so if you all remember in one of our previous lectures, we talked about what happens to the entropy of the system, right? When a molecule is placed in water molecules in a physiological environment, all of those water molecules will align themselves around this molecule. And when this molecule reaches its receptor, so the receptor, the water molecules that are associated with the receptor are also disturbed, right? So there we talked about how this molecule will have a hydrophobic core and a hydrophilic exterior, right? So uh, the physicochemical properties which talk about these terminologies are SASA, FOSA, etc. Okay, so SASA is solvent accessible surface area. So how much of the area of the molecule the solvent can access? So that is SASA. FOSA is hydrophobic component of SASA. Okay, so it talks about the saturated carbon and attached hydrogen. So this should be 0 to 750. The value should be in between 0 to 750. And FISA is the hydrophilic component of SASA. Okay, SASA का hydrophilic component. So this is nothing but the nitrogen, oxygen, and non. This is non-heteroatom component. So this should be in between 7 and 330. Then there is again called as something called as PISA. PISA is the third component of SASA, which talks about the pi bonding, and the double bond and triple bond content of SASA. So that should be in between 0 and 450.